this event's public, it will be filmed, and the film will be posted online. So just need to make everybody aware of that.
Well, I don't need to tell you who I am since you just heard it all, but my wife says, tell them who you are, you need to be personal. I retired 10 years ago, and I buried Dr.Vrain. I thought I was done with research and all that. I just wanted to become a gardener, which I did. I became a gardener, and I'm now enjoying it. And the last 2 or 3 years, I guess I resurrected Dr.Vrain. So I'm on a mission. I'm on a mission to educate. I need to tell you a story. I need to tell you what I did 20 years ago, somewhat, and what I see, what I read these days, because I've been reading a lot in the last few years, so in the last 10 years, I've been reading a lot, and I'm getting really concerned. And that's why I'm here, and that's probably why most of you are here. So I'm just going to show you some slides and give you a few facts, and it's going to take probably an hour or so, and then I will be totally open for questions. If you have any questions or concerns, I'll be happy to address you or give you my opinion. I give you the title The Gene Revolution because it was a revolution, it still is a revolution. In the last 20 years, it happened in agriculture. Agriculture has been completely transformed, revolutionized by this technology. You can see the lines going up. It started in 1996. 1996 is the first year that the first engineered crops were commercialized in North America, in the US, and then of course in Canada very soon after. So these curves are for the USA, but the same thing would happen in Canada, and there's a handful of other countries also where the engineered crops are very, very big. Brazil, Argentina for soybean, and China and India for cotton, Canada has canola and corn, and of course, and soybean, and etc. But those curves, I don't know if it's possibly too small for you to see, the pictures should be bigger. The curves tell you that basically more than 80% of all corn in North America is engineered. More than 90% of soybean in North America is engineered. Cotton in the south of the US, of course, and China and India, as I've said already. And there's a good reason for the success. This is an incredibly successful technology, whether you like it or not. Why did the farmers adopt it so much? Well, there's a good reason for it. Imagine that you're a farmer or a gardener. If you're a farmer or a gardener, the weeds are your number one problem. Weeds are overwhelming in the garden and in the fields. As a farmer, you spend more than 80% of your budget on pesticides, on weed control. All over North America, 80% of all pesticides are herbicides. It's not insects or fungi or nematodes or other pests, it's weeds. 80% of all pesticides are herbicides. Imagine a company that would come and say, we're going to take care of your weed problem for you. We have this magic product. It's completely innocuous, it's perfectly safe, it's like water. It's safer than aspirin. And you don't need to worry about your weeds anymore, you just let your weeds grow in the fields. And when you're ready to plant, you just plant your seeds, never mind the weeds. And when the time comes, you just spray this magic herbicide. And the weeds disappear. All your vegetables in your garden are standing up, there's no weeds. And all your crops are clean, there is no weeds. It's magic. It's not quite exactly like that, of course, the herbicide is Roundup. And the technology is the engineering technology that you know of already. but it does feel like it's a magic herbicide. And that's why the farmers are completely sold. If you're not using this technology today as a farmer, you're wasting time, you're wasting your money, you're not doing well. This is an incredible technology. It saves you time, it makes your life simple, you don't need to worry about the weeds anymore, you don't need to know your weeds anymore, you just spray. So this is what I'm going to talk about tonight. I'm going to tell you a few words briefly, what GMOs are, what crops are they. I just mentioned corn, soybean, cotton, etc. So I'll repeat myself. I'm going to briefly also explain to you how they are made, how are they created. I'll try not to be too technical. And then I think most of you are here because you want to know how are GMOs performing. And so we'll look at the claims of the biotech industry. Are the engineered crops reducing pesticide? Are they safe to eat? Are they innocuous to the environment? Do they increase yield? These are the claims I've just put those claims of the industry in just 4 categories, but this is basically what they claim. So we'll go through that. So let's start. What GMOs are. And I just gave you the story already. I'm ahead of myself. I gave you the story about the magic herbicide already. And it's very real. And this is why the technology has been incredibly successful. There's nothing for the consumer in it. Nothing at all. It's all about helping the farmers. And you may have read claims from the biotech industry that they're going to feed the world, and there's new crops in the pipeline, and there's drought-resistant crops, and this is all not real. The engineered crops are herbicide tolerance, HT, herbicide tolerance. You spray, and the crop is still standing, it was not hurt. 80% of all engineered fields, 80% of all engineered crops in the world are HT, herbicide tolerance. So huge success. And then there is another, this is Roundup Ready, of course. There is another technology, it's called BT. And BT stands for Bacillus thuringiensis. It's a big long name, it's a Latin name of a species of a bacteria. It's a soil bacteria. It lives in the soil, it's quite common. And this bacteria, when life becomes difficult, it sporulates. It just shrinks inside of its skin and it goes dormant to wait for a better time. More food, maybe more water, something. But when it sporulates, it also makes a protein crystal. And that protein crystal is incredibly toxic to insects and a few other organisms. There's a whole family of that bacteria, and they make different proteins. And the proteins are very specific. Some of the proteins are noxious to, very toxic to caterpillars, or butterflies, or other insects. And of course you can put the two technology together in the same plant. You can have corn that has been engineered to resist a herbicide and to kill the insects. So if you have on your farm a problem with insects, well, you can have both. You can have resistance to the insects and you can have resistance to the herbicide. Your life as a farmer becomes a lot simpler. Stacked again also in cotton. And this is sweet corn. And sweet corn has been engineered recently, and it is the first crop that is directly for consumption by humans. All the corn that's been engineered feeds cattle, or is for biofuel or for industrial processes to make vitamin C and to make high fructose syrup, corn syrup. But sweet corn is now engineered to be herbicide tolerant and to be resistant to the insects. And that's about 2 years old. It is now in the stores. And I did not buy sweet corn last year because I don't want to eat GMOs. Canola is a big crop in Canada. Almost all of canola in Canada is engineered. Herbicide tolerance. Flax is engineered also. We lost our market to the European Union and to Japan because of contamination of flax. Sugar beet. Sugar beet is a recent addition. I think it's 7 years old. And in 7 years, from zero sugar beet engineered, over 95% of all the sugar is now engineered. So when you buy sugar in the store, it is engineered. Alfalfa has been in the US for several years, and it is not quite in Canada, but it's due to be registered either this year or next year. And a lot of people are very concerned about alfalfa because alfalfa is very important for feeding cattle or for other uses, and the conventional and organic growers are very concerned, and they should. So that's what GMOs are. Basically, most of the major crops have been engineered, and this is the status quo today. So how are they created? Well, there's two kinds of technologies to create engineered crops. The first one is called a gene gun. It looks like a hairdryer, but it's a gun. It looks like a Star Trek phaser. But instead of shooting whatever Star Trek phasers shoot, it shoots little pellets, tiny little microscopic pellets of gold or platinum, something that will not oxidize, but have been coated with the DNA that you want. And then you shoot those pellets into the plant, into plant cells. Millions of pellets. And some of those pellets are going to find their way into the nucleus of a cell. And by some miracle, and we don't know how it works, the DNA that you on the pellet, that you coated on the pellet, the DNA will find its way and be integrated in one of the chromosomes of the nucleus of that particular cell. And that cell is now engineered. And all you need to do is to select that cell, because you're shooting millions of pellets and you're shooting millions of cells, and it's a very low rate of success. You have maybe a few successful events, only a few cells have been engineered. So you need to know which ones they are, because it's a huge amount of work to go from that particular engineered cell to regenerate a whole plant that will be engineered. So you need to know which events were successful. And to do that, you add to the gene you wanted to, like it could be the HT gene or the BT gene, you add a gene for selection. And for selection, normally, you use antibiotic resistance. Antibiotics kill bacteria, antibiotics kill plant cells, antibiotics kill all kinds of cells, even human cells. And so you are going to put all the cells that you shot, you put them on a petri dish that contains antibiotic. And the cells that have been engineered will survive because they are now producing antibiotic resistance on top of the HT or the BT genes, and everything else dies. So the antibiotic resistance is absolutely essential to the process. And I'm taking my time to explain this to you because it has consequences. So this is the gene gun technology. And there's another technology, and for that you use a bacteria called Agrobacterium tumefaciens. It's a very long name. You don't need to remember it. It's a bacteria also that's very common. It lives in the soil. And we learned about 30 years ago that this bacteria is we knew it was a parasite, but we learned how it does its parasitism. And basically this bacteria is the original genetic engineer. It's a perfectly natural process. This is what it does. It's a bacteria, and like all bacterial cells, it has one small chromosome, and it also has some very tiny mini chromosomes that are usually circular, so circular DNA called plasmids. It's not very complicated. Don't pay attention to all that. It's a love story. I'm going to tell you a love story. The bacterial cell in the soil swims to the plant. It's attracted to the plant. The plant is there. It's like us, you were doing pheromones. The plant is continually excreting sugars and chemicals to say, hey, I'm here. And the bacteria swims to the roots. And it contacts the roots. And it's talking, biochemically speaking. It's talking with the plant, with a particular plant cell that it came to. It said, hey, I like you. Can we get married? And the plant cell doesn't really have much choice. And as this conversation is going, the bacteria inside the bacteria, that little plasmid, that little round plasmid that I showed, it's the TI plasmid that you see here. Well, the TI plasmid is a round piece of DNA, and it has all the genetic information that's necessary to actually do the engineering. It has a piece of DNA which is called a tDNA. At the bottom, I don't know if you can see that. It's called a transfer DNA. And then the rest of the plasmid is a whole number of genes that code for many proteins which are called the virulence proteins. And these proteins are created, are formed, as soon as the bacteria comes to the root. And these proteins are now going to cut off the tDNA, take it out, or actually make a copy of it. And then they're going to grab that DNA. Now you have to picture this. I don't know if you know this, but proteins are the molecules that do the work in the cell. Proteins can move. Proteins have a kink, and they can be in one conformation or another. They move. Sugars, carbohydrates, and lipids do not move. Proteins move. Proteins make life. Life is movement. And that's what these proteins are going to do. They're going to take that piece of DNA and they're going to move it through the cytoplasm of the bacteria, and then through the bacterial wall. They make a bridge, a little green bridge at the bottom, and they're going to take that piece of DNA and push it through that bridge, and now they're into the plant cell, and they're going to move it across the plant cell cytoplasm and into the nucleus of the cell, and integrate that piece of DNA into one of the chromosomes of the plant cell. The plant cell is now engineered. And this is what this bacteria does for a living. And now that the plant cell is engineered, it contains the tDNA. And the tDNA basically says, now you're going to do what we want, the bacteria is speaking, and what we want is to be fed. And the plant is now going to feed the bacteria its favorite food. And the plant cell is going to make bacterial sugar that do not exist in the plant kingdom. Opines, they're called. The bacteria is basically, remember, they wanted to get married? The bacteria is basically making the plant feed it. It's extremely elegant, it's pure parasitism, it's beautiful. Once you know how this works, if you are a genetic engineer and you know enough molecular biology and you have the tools, by the way, are proteins. They're enzymes. We cut the DNA, we cut and paste, we do all kinds of things with proteins. And you can do that inside the bacteria. You can replace that tDNA with the gene you want and that antibiotic resistance gene I talked about. And you can put it in the bacteria, and the bacteria is asleep, it doesn't know, it doesn't feel a thing. And when the bacteria comes to the plant, now it's going to push into the plant the gene you want with the antibiotic resistance gene. And you have now engineered the plant to do whatever you want. Would you like to make a human protein, insulin? Would you like a corn plant to produce insulin? Easy. Would you like a soybean plant to produce lysozyme? Any protein, you can do that. This is how engineered crops are created. The next step, of course, is you have one cell that has been engineered. It's called an event. And you have to regenerate a whole plant from that. So it's a little bit complicated. There's a lot of plant hormone involved. It's called tissue culture. That's quite a specialization on its own. But you can regenerate a whole plant from that particular engineered cell. And what you have now is an engineered plant. You just multiply the plant like you normally would, and now you have fields of engineered crops. Hundreds of millions of acres of engineered crops today, created this way. So do they deliver? And what do they deliver? Well, the first claim of the industry is basically these crops, when they were invented 20, 25 years ago, when I was working in that field, that was a green technology. We were going to reduce the use of pesticides. And that's definitely the number one claim of the industry. These crops are there to reduce the use of pesticides. Well, it kind of happened with the BT technology, the bacteria, the pesticide technology. The plant has been genetically engineered to produce the protein that I mentioned. Every cell of every plant is producing that protein and is toxic to insects. You don't need to spray anymore. So that technology has been successful in reducing the amount of insecticide. Not marginally. I think from 96 to 2011, there was like 100 million pounds of insecticide in reduction compared to what was before. It's not negligible. But there is a phenomenon that was predicted as soon as the crops came in 1996. The entomologists, the people who specialize studying insects, noted that basically the insects are perfectly capable of overcoming the pesticides that we throw at them. You are using a pesticide or an insecticide, you spray your crop with an insecticide to kill a particular insect, and almost all of them die, but a few of them do not die. And then the next year you do that again, and for some reason more of them do not die. And the ones that do not die are resistant to the insecticide. And they basically replace the original population, and very soon, in a few years, you have a population of insects that are completely resistant to that particular insecticide. It's time to switch to another chemical. And the chemical industry likes that. And that's what's happened with this technology. The weeds have become resistant. In a few years, they've become resistant. Some weeds became resistant. And in the last 10 years, more and more weeds have become resistant. Today, we have about 40 species of weeds that have become resistant to Roundup. And about 50%, half of the acreage in the USA is now infested with one or more of these resistant weeds. It's a very serious problem. Remember what I explained earlier when I started? The weeds are overwhelming. The weeds are number one preoccupation of the farmers. This technology became so successful because of that. And now we're back to step one, where the magic does not work anymore. And we don't have resistant weeds in BC yet, but it's a question of time. It's biology. 40 species of weeds. They're in Ontario and Alberta, not yet in BC, but it could happen this year or next. And the numbers are climbing. Basically 85 million acres of cotton, soybean, and corn are infested with weeds. And the only thing that the farmers can do is to hire people to pull the weeds by hand. The herbicide does not work anymore. Or use a new technology that has been in the wings because the chemical industry, the biotech industry are the chemical industry, they're the same. And they're in business to sell pesticides, to sell chemicals. So the herbicide tolerance, which was successful in the beginning, is now not completely useless, but useless in half of the fields. And the use of the herbicide has shot incredibly. So there's definitely no reduction in pesticide, quite the opposite. And the biotech industry knew this was coming, so the last few years they have developed a new technology. It's still HT, herbicide tolerance, but let's replace Roundup with another herbicide. So now we have 2,4-D. Some of you are old enough, many of you are old enough to remember the Vietnam War. 2,4-D was Agent Orange, contaminated with dioxin. So obviously 2,4-D is not contaminated with dioxin today. We're not in Vietnam. But there are indications that it is actually toxic, quite toxic, to many life forms, including humans. The insecticide technology also is failing because the insects are becoming resistant very quickly. And so now again, the magic is not working. You can pay a great deal to buy these technological seeds from Monsanto and plant your crop, but the insects are resistant, the weeds are resistant, and so now the farmers are told to spray insecticides on their leafy crops to avoid either to kill the insects that are resistant to the technology or to avoid the insect becoming resistant to the technology. Either way, the farmers are losing. So I think the case is made that GMOs do not reduce pesticides. Absolutely not. Quite the opposite. Quite the opposite. And GMOs increase yield. Well, that's another claim by the industry, and there is nothing in the technology to increase yield. The technology is very simple. The technology is to make the plants resistant to herbicide or make the plants insecticidal to kill insects. There's nothing about increasing yield. If you have good insect control, yes, you will have an increase in yield compared to what you would have if the insects were destroying your crop. Yes. But to increase yield compared to conventional crops, no. There is no increase in yield. Actually, the opposite happens. There are a good number of academic studies that were done in the USA, Kansas State University and Wisconsin, that show that there is what is called a yield drag, that the technology, the insertion of the herbicide tolerance gene is actually costly to the metabolism of the plant, so that the plants yield less. And the numbers are about 5 to 10% less. There is no increase in yield. A publication came 2011. It was 2009, updated in 2011, and showing data, numbers, statistics from the USDA, from the US Department of Agriculture. These are the yields that we have in the USA. There is no increase in yield. There is no argument. And Monsanto admits that. No reduction in pesticide, no increase in yield. The next claim is this technology is completely innocuous, there is no effect on the environment. And I'm just going to address two issues with that claim. The first one is called contamination, the next one is called genetic pollution. Contamination means that if you are a farmer and you have a field of genetically engineered corn and your neighbor does not grow engineered corn, he grows conventional corn or perhaps even organic corn, you can be sure that the pollen from your crop is flying onto their field. It used to be that in agriculture, if I was a farmer and you were a farmer and my cows went into your field to feed on your crop, you would come and knock on my door and say, your cows are in, I did again. Oops, sorry, it was my responsibility to come and get my cows and maybe even to put a fence to avoid damage to your crop. And that's not the way it is anymore. Don't have to, because 90% of people growing corn grow engineered corn. You're a very small minority. Tough. That's contamination. You cannot grow a regular crop next to an engineered crop. You will have contamination. The pollen flies. Doesn't matter if it's corn or soybean or canola or alfalfa. Contamination. Some growers, conventional growers and organic growers, lost their market to the European Union in Canada. Hundreds of millions of dollars worth of market because of contamination. Same thing with flax. Contamination of flax. We lost our market. And Phil, the previous speakers, mentioned wheat. There is no engineered wheat in North America. The growers decided to do without, because more than half of the crop of wheat in North America is exported to Japan, to Europe, to China, and they don't want engineered wheat. And in Oregon, 2 or 3 weeks ago, a field was discovered in Oregon with engineered wheat. And the only way that can happen is that that came from trials from Monsanto. 10, 15 years ago, Monsanto had thousands of trials in Washington State, in Canada, everywhere, just to try the crops, just to see how it would be. And the plants have kept going since. And now we find them. And as Phil said, Japan immediately, Japan, Russia, Korea, and probably a few other countries today have said, we don't want US wheat anymore. So they're about to have a taste of their own medicine. Not very good. The next thing, I'm taking you to China. The next issue is called genetic pollution. And contamination is when the pollen flies. Genetic pollution is a little bit different. Genetic pollution is lateral gene transfer. Gene transfer is when genes go from one organism to another. All of you have done that. You call it sex. Sex is vertical gene transfer. You give your genes to your children. Last time I mentioned sex, I heard, yoo-hoo! Lateral gene transfer, bacteria do that all the time. Lateral gene transfer is when you give your genes to the person sitting next to you. That's what bacteria do. When bacteria have a gene that is useful, it can spread like wildfire. Remember what I talked about the antibiotic resistance? That could be useful. Well, the Chinese did a study. This is published in Sichuan University in China last year. They sampled rivers. They took water samples from 6 rivers in China, looking for antibiotic resistance. And they found antibiotic-resistant bacteria in all the samples they took. And the antibiotic resistance came from a synthetic gene, a gene that was incorporated into bacteria in the lab, not natural bacteria. We did that. Somebody did that in China. And whether it came from a lab somewhere or whether it came from engineered crops locally is not known yet. But it's a real concern. You now have antibiotic resistance completely out in the open, and the bacteria are happy to share it all the time. And you know how the medical establishment is concerned, and we all should be concerned, that we are losing our antibiotics because the bacteria are becoming more and more resistant to more and more antibiotics. It's a serious concern. The transgenes, this is the genes that we put in the engineered crop. The transgenes can spread from transgenic crops to soil organisms, to the bacteria in the soil. And apparently they also can spread to the bacteria in your guts. There are several studies that show that if you eat engineered crops, the bacteria in your intestine, who are extremely numerous, you may not know that, but 10% of your cells are human cells. 90% of the cells in your body are bacteria. We have an incredible number of bacteria in our intestine, and they do all kinds of things, useful things. If you eat engineered crops, the transgenes find their way into the bacteria in your guts. This is documented, there is no argument. Lateral gene transfer. There is a good possibility that you are becoming an antibiotic resistance factory, or a pesticide factory for that matter. So, I don't think that engineering technology is innocuous to the environment. Contamination is a very serious problem, and genetic pollution is probably even more serious. Somebody wrote a long time ago that it probably would be a bigger concern than nuclear radiation or some such. Well, at least they are safe to eat. So that's a bit of a story too. There is a concept, it's called substantial equivalence. When the technology was invented 30, 40 years ago, and when the crops were grown, the US government And the federal agencies, the Food and Drug Administration and the USDA, the US Department of Agriculture, coined the term substantial equivalence, meaning this corn is engineered, and so yes, it does have a gene from fish or bacteria or whatever, it doesn't matter. But it does look like corn, and it tastes like corn, and it grows like corn, and there is no difference. It is equivalent. And with that phrase, substantial equivalence, that means we don't need to test it. We don't need to look at it. It's the same. There is no difference between other varieties of corn and engineered corn. And that goes for all the other crops. The FDA wrote a letter, and for every crop writes a letter to the biotech companies saying, your crop is substantially equivalent, we don't need to look at it, we're not even interested in doing any research on it, you don't need to do any research on it. In 1996, remember, that was the year when the first crops were commercialized. And before the crops were commercialized, the scientists of the Food and Drug Administration were all together. There was a huge consensus. They all predicted that this was a dangerous move, that those particular crops could definitely be dangerous and toxic and could create allergens. The engineering of the crops could create allergen, could create new proteins, I call them rogue proteins, new proteins that could be allergenic or toxic, could create new diseases or nutritional deficiencies, etc. This is documented, this is published. 1996. The administration ignored them and went ahead and accepted the crops, substantially equivalent. In 1998, I was in Scotland. I was a scientist. I was in Edinburgh for a week, and for a conference. And during that week, there was an incident. A scientist at the Rowett Institute in Aberdeen in Scotland went public with research. He was testing GMO crops, and he found them to be very toxic. And rather than wait and publish his results, which is what you do in science when you're a scientist, first you publish, then you can talk. Well, he didn't do that. He went on television and he made public a statement that no, he wasn't going to eat any, and actually his rats were damaged. His name was Arpad Pushtai, Dr. Arpad Pushtai. He was at the end of his career. He was one of the top scientists in the world in toxicology. He had €1.5 million grant, a huge grant, to design a protocol for the European Union to test genetically engineered crops. Since the European Union government knew very well that the US was not testing them, or Canada was not testing them, they were not going to accept them without doing some testing. So they asked him to put together a test, and that's what he did. He fed rats for several months. He fed rats engineered potatoes that he had designed. And then he sacrificed the rats and looked inside. And he found organ damage. He found the liver, the kidneys, the intestine, the brain, the testicles were not normal. And he published his results in probably the most prestigious medical journal in the world, The Lancet. And in 2000, this is 1998, and published in 1999. In 2002, the year I retired, was the completion of a huge project. It's called the Human Genome Project. It took 10 years and a lot of knowledge to engineer, to decode the whole genetic code of the human being, all of it. Every gene of a human being was decoded, and we had that information. And we have known for a good number of years now, everybody is pretty much in agreement that the human body functions with approximately 100,000 proteins. Our body contains 100,000 different proteins, most of them enzymes. They do the work. This is life. Every time you move a finger, there's proteins working to do the movement. And the paradigm until 2002 was from a hypothesis that was advanced, that was put forward 70 years ago, in the late 1940s. Before Watson and Crick, before we knew what the DNA looked like, that basically a gene would code for one protein. Each gene codes for one protein. And then 60 years later, we have this project that says we have 100,000 genes, and oops, we only have just over 100,000 proteins, and we have just over 20,000 genes. A human being has 22,000 to 23,000 genes and 100,000 proteins. It's a lot more complex than we know. Which means that, in a nutshell, we don't know what we're doing. Each gene codes for several proteins according to the weather, according to the environment of the cell. Well, I'm going to make this protein because this is what's needed now. Oh, now we need a different protein. So there's a lot more than what we know. Each gene makes several proteins. They're pretty. These are models. And this is a Western blot. A Western blot is just, some of you have been to high school and you did some electrophoresis, etc. It's the same thing. And these are protein bands. So when you look in an engineered plant, if you have a Bt plant, you should have one band for the Bt protein, one. Because you have a Bt gene, you expect one protein. Oops, there are many proteins. Well, one way to deal with that is to ignore them, and that's what the industry and the government agencies are doing. It's just background noise. They're not really there. Moreover, the engineering technology is really blasting a gene into the genome, and we don't know where. We have absolutely no control where it goes. So the biotech industry loves to say that genetic engineering is really precise, and it's exactly the opposite. It's incredibly messy. And we don't know where the gene goes, and we don't know what the proteins will be. And this is very real, and there are many publications today that show that the proteins produced by the genetic engineering technology are much more than what's expected and quite different. It's all published. All the publications, I'm just going to show you a whole series of publications. These are scientific publications in peer-reviewed journals, most of them out of Europe and Russia and a few other countries, because there is no research done in North America unless the biotech industries says yes. You can do research on our product, but it's secret. We have to see the data first before you publish it. This is out of Belgium. GM crops have different proteins compared to the original proteins reported by the companies. The proteins are just not what is expected. Journal of Agriculture and Food Chemistry. Some scientists took a protein from bean, totally safe, and engineered it into peas, and now the protein is toxic. And before the engineering process, it was not toxic at all. And most of you know that there is some kind of controversy. There is a debate. There is research done by the biotech industry. Actually, the biotech industry does very little research, but they give a great deal of money, they grant a great deal of money to universities to do research on their product. Very little about safety testing, but a lot of money goes to universities, mostly in North America, but also somewhat in Europe. And most of the publications coming out of those grant money, say that the technology is safe and innocuous. And there is another body of research done by other labs without grants from the biotech industry, done by government agencies in Europe, that show exactly the opposite. That the technology is actually not safe, that the proteins are not safe. Some of them are actually quite allergenic or toxic. Remember I said the Food and Drug Administration scientists in 1996 predicted that the technology would create growth proteins, some of them would be allergenic, toxic, etc. And this is exactly what we're finding. The BT protein is actually quite allergenic in rats. This is all in rats. If you see a rat in a corner, it's rat research. The rats are the canary in the mine. You remember the miners 100 years ago? They would have the lanterns and the canary cage, because the canary would be singing until the methane became so much that they would die and there was going to be an explosion. When the canary stopped singing, it was time to get out. The rats are the canary in the mine. BT corn is allergenic. BT corn causes anaphylactic response, Journal of Immunology. These are international journals. Allergenicity of proteins in engineered foods. Hematotoxicity of BT proteins in mice. Damage to blood cells in mice. Mice fed GM soy have damaged liver. Rats fed Roundup Ready corn have damaged liver and kidneys. This study is a recent study and it lasted for 2 years, the whole life of the rats. Mice fed GM soy have damaged testicles, uterus and ovaries. European Journal of Histochemistry. And then there is another aspect to it, because Roundup, the herbicide, remember when I started I said it's like water, because that's what the Monsanto, I was going to say the biotech industry, but Roundup is a Monsanto product. That's what Monsanto researched and affirmed and basically publicized 30 years ago, that Roundup was absolutely innocuous, had no effect on the environment or anything other than the plants. Roundup, when it was patented, was a chelator. It was not a herbicide, it was a chelator. A chelator is a molecule that grabs onto metals and basically takes them out of commission. There's quite a few of them. But Roundup is an incredible chelator because it does not chelate, it does not grab onto one kind of metal, it grabs many different kinds of metals. So when Roundup comes into a cell, a plant cell, a bacterial cell, a human cell, any cell, it grabs onto metals. And it so happens that many of the proteins that do the work, well, they have a metal atom as a cofactor. Some of you may be familiar with hemoglobin. Well, hemoglobin has a metal in the center. Or chlorophyll, there's a metal atom in the center. And Roundup prevents that metal from working because it grabs onto it, so that the cell is not working as well. That protein is not working as well, or not working at all. It's a very insidious process. It's not incredibly toxic that the cell dies immediately. No. It takes a long time. And we are only discovering in the last few years that Roundup is actually quite toxic to cells, including human cells. We knew that it was teratogenic. It creates birth defects in frogs. That was shown 20 years ago. But frogs, we need a good herbicide in agriculture. What about frogs? Never mind. Now we're talking human cells. Cytotoxicity on human cells of Bt proteins alone or with glyphosate. Glyphosate is the scientific name for Roundup. It's the active ingredient for Roundup. Glyphosate, Roundup, induces apoptosis, which is a jargon for cell death. Basically, the cell commits suicide. That's what apoptosis is. So Roundup kills human cells, umbilical cells, embryonic cells, placental cells. Roundup-based herbicides are hormone disruptors in human cells. And again, all this from grabbing onto metals. It's very simple. Cytotoxic and DNA damaging properties of Roundup in human cells. The American Academy of Environmental Medicine, these are doctors, MDs, they can read. And they are just as alarmed as I am, and that's what they wrote several years ago. Based on independent studies showing infertility, immune dysregulation, your immune system is shut down. Accelerated aging, I don't want that. Dysregulation of genes associated with cholesterol synthesis. Insulin regulation, you get the picture. The American Academy of Environmental Medicine suggested a moratorium on GM foods, testing and labeling. Again, this is a very large group this is an international body, and these are all MDs. And I'm just finishing here, I have no idea what the time is. There is this publication, GMO Myth and Truth. It's available online. It is a compilation. A lot of what I just said comes from this publication, or at least it is in that publication. It is a compilation of scientific reports, government reports, published scientific studies in international journals, just like I showed you. This was compiled by a genetic engineer in London, in England, and this was published a year ago, in June 2012. And it is very easy to read because it was written by a lay person, a journalist, so she translated the scientific jargon, so it's very easy to read. It's available online at Earth Open Source, and it's free. You can download it for free. It's about 120 pages. It's very good information. So to finish, I would say that with all the problems that this technology has, the super weeds, the weeds that are resistant, the superbugs, the insects that are becoming resistant, the contamination problem I mentioned, the genetic pollution, which is very serious, the allergen, the proteins that are allergenic, the proteins that are toxic, and and whatnot, and I didn't put a big dark cloud for Roundup, but it should be there. And my conclusion is that the future of agriculture is not necessarily engineered. Thank you.